Welcome to the UC Riverside College of Natural and Agricultural Sciences 2022 Science Lecture Series on Big Data Science. My name is Madeline Haddad. I'm a second-year math major here at UC Riverside, and I am a CNAS Science Ambassador. Before we get started with today's presentation, we at UCR would like to respectfully acknowledge and recognize our responsibility to the original and current caretakers of this land, water, and air, the Cahuilla, Tongva, Luiseno, and Serrano peoples, and all of their ancestors and descendants, past, present, and future. Today, this meeting place is home to many indigenous peoples all over the world, including UCR students, faculty, and staff, and we are grateful to have the opportunity to live and work on these homelands. And now, I would like to bring up CNAS Dean Catherine Girke.
Thank you, Maddie, for that introduction. And thank you to all our guests in the room, as well as who are joining us virtually on Zoom. So welcome to the 2022 Science Lecture Series on Big Data Science. Each year, the CNAS Science Lecture Series takes on topics that impact us all. Many of you can relate in your everyday lives how technologies collect and analyze data in various ways that we had never would have imagined. So the big data that those technologies generate have transformed not only how scientists do their research, but also how we view our world. Specific to our lecture this evening, Dr. Thomas Girke's presentation on big data at the intersection of drug discovery and genome biology couldn't be more relevant. But before we bring up Professor Girke, I'd like to introduce our moderator for today's presentation, Dr. Corinne Larocque. Dr. Larocque has more than 20 years experience in parasites, from drug discovery, cell cycle, and functional genomics in eukaryotic organisms, with a particular interest in Plasmodium, the protozoum that causes malaria in humans. Dr. Larocque's research focuses on new high-throughput functional genomics methods. In particular, her laboratory develops new high-throughput assays to identify epigenetics, chromatin, and molecular features driving parasite gene expression. Her expertise in drug discovery and molecular biology techniques, as well as her in-depth experience acquiring, handling, and analyzing large genome-wide data sets from a wide array of samples and techniques, have been demonstrated with over 100 peer-reviewed publications, 14 book chapters, and 1 U.S. patent. That's quite a mouthful. I'd say in brief, she studies parasites and infectious diseases. So with that, she's also the director of the UCR Center for Infectious Disease and Vector Research, a center of 35 faculty that incorporates genomics and systems-based methods to find new approaches to control the spread of infectious diseases in general and diseases vectored by arthropods. Again, a world-recognized expert in parasites and infectious diseases. So please join me in welcoming our event moderator, Dr. Corinne Larocque.
Thank you very much for this very kind introduction. It's my turn to actually introduce our speaker of the day, Dr. Thomas Girke. Professor Girke is a professor of bioinformatics in the Department of Botany and Plant Science at UCR. He's also the director of the High Performance Computing Center and the director of the Genetic, Genomic, and Bioinformatics graduate program. He received his BA, MS and PhD in biochemistry and molecular biology from the University of Hamburg in Germany. He then did his postdoc in Michigan State University, where he developed one of the first DNA microarray study to understand transcriptional regulation and development in Arabidopsis seeds. It was in the early 2000s, and that was a very exciting time to start a genome-wide type of study. So he then decided to do a short jump into industry, where he directed several computational research projects in agricultural high-throughput screening assay for Don Agroscience and the Don Chemical Company, but then decided to come back to academia. And in 2002, as a bioinformatic and coordinator of bioinformatic facility and professor of bioinformatics, he came back at UCR. And then at UCR, he developed impressive, innovative, and computational biology and drug discovery research programs that were funded by large grants from a variety of institutions. variety funding agency, including NSF and NIH. He has published over 77 publications of iJournal Impact, and last but not least, as the director of the UCA High Performance Computing Center, is enabling big data research for over 150 groups from all school and college at UCL. So we're looking forward to hearing about big data at the intersect of drug discovery and genome biology. So I will take questions after Professor Girke's presentation from the room here or from the online attendance. So if you have any question, please write down your question in the chat box, and I will read this question after Professor Girke's presentation. So again, thank you, and I'm welcoming you for your big presentation on big data.
Okay, thank you very much, Corinne, for the nice introduction, and thank you to the organizers, SINA's Science Lecture Series, Dean Ulrich, and everyone involved, and working hard to make this possible. And so, today I will present pretty much my view on big data, how we work with big data in my scientific area. And I will provide an introduction that will first provide a little bit background from someone who works in bioinformatics and computational biology as to how we understand what big data are and how we use big data. And so the first part will be a little bit more general. And then I will provide an introduction as to where the data comes from in our area, and where the big part of the data comes from. And then also a little bit about the infrastructure that is important to us to deal and handle big data. How is this possible in a university setting? And then I will provide a couple of research examples that relate more to my research. So the first part is a little bit more general for coming from an area like myself, people working in this field of research. And then I go a little bit more specifically into research areas that relate to my research program. So first of all, I will mention a couple of challenges with big data and how to actually utilize them. And from that perspective, one could also view big data as a challenge that is actually obstructive to sciences. And but I will hopefully be able to convince you that it's actually a great opportunity to advance sciences, and it's exactly the opposite. Yeah, but there are some challenges, and how to overcome them, I will provide quite a few examples, often related to the infrastructure part, and hopefully convince you that it's right the opposite. And then when we work with big data in different fields, the way we address this can come from two different perspectives. And in my area, we actually work always on two main, come from two more main areas, and this can come in every area, like in engineering or in biomedical sciences where big data is used, or in agriculture. People can either work on method development to handle and process big data, so pretty much on methods to analyze big data very often. And as you will see later on, there's quite a few projects where we developed algorithms and software for analyzing big data. And this can happen in many, many different areas, in particular statistics and many engineering disciplines, including computer sciences, work in this direction as well. And then you can come from a more discovery-oriented perspective to work with big data and actually use big data to make new discoveries. And then we have to understand the properties of big data, what new opportunities they provide to us, what we could not do with small data or traditional data that we had before. And many examples there, I will provide quite a few of them, are in high-resolution environmental sciences or high-throughput genomics, like Dr. Laroche is working on, precision agriculture, or like also very exciting fields like in personalized medicine and many other things. So it can be either, or it's often the combination between developing tools for analyzing the data and then using these tools to make discoveries and able to make those discoveries. And we do both of them. And then the type of new discipline that is often coming along or is joined into this area of big data is often called data sciences. So there's many programs, also here at UCR, master programs and also PhD programs, where this term comes up, and it's very, very popular. There's many master programs at many, many universities, as well as PhD programs in data sciences. And when it comes to big data, then you can immediately also call someone who does data sciences a big data scientist. And the simple definition here for me, what a data scientist actually is, or what data science programs are doing, it's a multidisciplinary discipline for us that combines statistics as well as computer sciences that relates to data analysis, and also includes at least one area that is experimental, or where the data is coming from. When it's maybe related to finances, it's not necessarily experimental, but a more applied discipline. Some people also call this driver areas. And the combination of this makes this multidisciplinary. And usually data scientists are also users of data who care for the data. They like to learn something from the data. And that is a important objective within these disciplines. And I guess in my field, when I teach students about big data analysis, then it's often relatively related to those three components, as well as because we work always in certain types of biomedical areas or agricultural areas, we are certainly interested also to drive the sciences, the discovery-related sciences as well. So this is a combination between engineering and building something, as well as making discoveries to understand nature. Yeah. Okay, and then a little bit about, I guess it's a little bit about the definition of big data relative to its size and what are the properties. So very often you will see these 3 V's about big data, what defines them relative to traditional data. And so that it's often the data volume is much larger, yeah, so the size of the data is larger. Then often they are more complex, so it's not always that the volume is so tremendously larger, but often more complex, so that we have to have new methods to analyze more complex data sets, integrate them, and so forth. And the data comes also with higher, at a much higher rate, meaning per year or time unit, we generate more and more data that also increases the size of these data. So that defines those 3 V's, volume, variety, and velocity. And then the size of the data sets is relative to when it was collected. So, and also that's pretty much the history of our computer systems, how we are able to actually store data. Big data was always around. We could take images, but we couldn't store them in the past. And nowadays we have computing systems available. In 2000, around 2000, big data might have been in the size ranges of gigabytes. In 2010, it was in the size range of terabytes. And now at 2020, we are more in the petabyte range. And there's also exabyte data sets. There are facilities dealing with this. But usually you have to have an entire big server room that is 4 times the size of this room, filled only with hard drives to actually store this much data. Yeah? So if you have a petabyte, for instance, to put this in perspective, it's roughly 10,000 laptops, the storage space on 10,000 laptops. And then with respect to the opportunities, and that goes more into this discovery-oriented area of big data, it is really trying to utilize the big data and find new opportunities to make discoveries that are only possible because of these data sets, that we can identify patterns in data and make discoveries from these large data sets that would not be identifiable in smaller data sets. I think that is one of the components to understand why this is relevant. It's not just the size, making it bigger and it's more interesting just because of the size, but there are new properties because of it that helps us to advance sciences. And then it also comes with challenges, and I guess that relates a little bit back then to the infrastructure I will refer to that is required to analyze data, big data, is the data storage you need to have in place to store it. When it comes to petabytes, it's quite expensive. Those systems often cost you $1 million quite easily. And you also need the processing power. So it's not just the storage and add one CPU to it that usually doesn't cut it. You need an entire cluster to attach it to it to analyze these big data sets. And then very often it's also, and that relates to the method development, more efficient algorithms to analyze these data sets because that sometimes even outperforms improvements in software, and algorithms can outperform improvements in CPU speed by many magnitudes as well. Yeah? But that often depends. So often we want both, more CPU, faster CPU, as well as better algorithms. And then the typical workflow when we work with these data sets, I guess that applies to all these different areas in physics, chemistry, agricultural sciences, and biomedical areas, is to first store the data, we manage it, put it into databases, that helps us organize the data, and then we want to analyze them. That comes then to these algorithms that we need and model the data. And then to take full advantage of these data sets, there is also a lot of heterogeneous data that we want to combine and integrate, and that provides new opportunities, and that's an additional component of analyzing these data sets. And that's what's very important to us often is to make efficient decisions in order to work with these big data sets. You need to have these large storage systems available. Usually you don't want to have them in your office. They wouldn't even fit in there. So you have to learn or have utilities available to work remotely with those data sets. And you have to have sufficient CPU power that is also then usually attached to those data sets. So very often we can either work in the cloud or we can work in big server rooms that we access remotely where the data is stored and as well the CPU is available in those server rooms as well. And then, yeah, algorithms, again, efficient data structures. And then because of the complexity of these analysis routines, there's many, many more steps often involved to deal with these data and analyze them. One wants to have protocols or procedures available that are actually reproducible. So the reproducibility for us is one of the most important components to allow us that actually what one scientist finds, another scientist can actually reproduce. And hopefully also at a different institution, yeah, or university. And that's a very important component there as well. So these were just these general perspective from someone who works more in the agricultural and bioinformatics field as well as in drug discovery in bioinformatics as to what big data is for us more formally. And now I show a few examples as to where the big data is coming from. And Karine will certainly relate to those technologies. For me, I'm more a user of these technologies nowadays because I'm entirely a computational scientist. So I don't run a vet lab where we do the dirty work. We pretty much let other scientists do this, and we do this through collaborations. So where does the big data come from? So there are, I think one of the big key drivers is DNA sequencing. I think that's the technology that came out first that accelerated in the early 2000s and in 2010 and now more and more so that we can actually sequence every single organism on this planet, as well as every single human in a full population. There are countries who are already pursuing this to sequence the genome of everyone in their population. And that is possible. There are big data banks like the UK data bank that warehouses huge amounts of biomedical data. Why this was easier to accomplish in Britain is something that we could discuss later on. Then it's here. It's often for regulatory purposes or reasons. and privacy reasons, of course, there are big concerns sometimes. But that is probably the area that has driven it the most. On proteins, it's often harder to sequence them. So we also want to get the sequence and modifications of proteins. And the technologies that they are very expensive, but the chemistry on proteins makes it a little bit harder to work with them than DNA, that is pretty much like a hard drive for us almost, that can replicate things and duplicate itself. But something like this is not built into proteins, unfortunately. And we call this proteomics, this area. And it's driven by certain technologies that are related to mass spec. And metabolomics is another area that allows us in our blood to identify all the molecules that are synthesized by metabolic pathways and so forth. And then in my area also high throughput drug screens. They're a little bit more traditional. They're just high throughput, but nothing new fancy in terms of nanotechnology. This one is highly driven by nanotechnology that has improved over the last years and developed by engineers in bioengineering here at UCR. They do similar things. And then there's, of course, many, many other technologies that contribute to this, like high throughput imaging and many, many other areas. So this is pretty much the source as to where the data comes from in our area. And then this is just for definition. I will often use this term, and you will hear it very, very often. There's the suffix here of ohm and omics. And it's a very simple thing to understand. Is usually you have something in a cell that contains the DNA of an organism. And in the DNA, all the individual genes are encoded. And we talk about all the genes that are represented in a cell. This is then the complete sets of those components identified here. And then the first one that was introduced was then the genome. And then we talk about every single mRNA or transcript in a cell, and we call this transcriptome. And when it's on the proteins, it's proteome. And then there come these technologies, the ones I showed you before. Often they are then called omics technologies. And all they do is often giving us the opportunity of coming from the high throughput or parallelization to analyze these molecules on a large scale so that we come close to a genome-wide level of analysis. So we often obtain a complete profile as to what is present in a cell. Yeah? And these are these omics technologies. I will not go into all these details, but I think it's useful and easy to understand as to why this term is useful. And then this is often used for things nowadays. By combining these different technologies, we often talk about multi-omics, and that means nothing else than using data sets from DNA sequencing as well as proteomics, as well as metabolomics, and many, many other things, high throughput small molecule screens or drug discovery, and combine then those different data sets through different analysis routines. So there are these different instruments that create those technologies. And these data sets are then naturally even larger and need to be stored and analyzed and integrated. And very often we do this through databases and also sharing the data publicly as often as is possible. With human data, it's often a little bit more difficult to openly share them. And then the new opportunities that are rising out of these technologies are, for instance, personalized medicine. That gives us the opportunity that someone can go who has a severe disease, and the physician actually can decide to sequence their entire genome. In cancer diagnostic, it's very often done when cancer samples are taken, that the entire genome of that cancer sample is being sequenced. And that provides many, many, much, much higher resolution insight into those diseases. As well as comparative genomics, that means that something I mentioned before, where we can sequence every single species on this planet, including even extinct species. If we have access to their DNA and it's still intact, we can sequence them. We can sequence entire populations. And then there's also technologies available that we call mixed population sequencing. People call this then metagenomics. That's often with microorganisms, where we take complex samples that are hard to dissect into individual entities of species, and then sequence in their entirety. And through algorithms, we can then dissect as to which species these individual molecules came from. And then another exciting area is then single cell technologies that are also available now to us. Okay, and so this was pretty much the different areas where the data is coming from. And to analyze these data sets, as mentioned in the introduction, I'm the director of our High Performance Computing Center, and for very selfish reasons, I'm bringing up this slide here as well, is that a lot of the analysis that we do, we perform on our clusters. And a compute cluster is nothing else than a huge amount of storage space where the CPU, GPU is directly attached. It's all warehoused in a big server room. And it helps us, first of all, to store the data and have the CPU directly attached to those data storage devices and to analyze them efficiently. Another important thing there is to share these data sets efficiently. Is that many people can work in parallel on the same system. So you're not, making multiple copies over several computers. You have it available, centralized, and hundreds of people can work simultaneously on those systems. Okay, and then the next part is diving into more the research that we are doing, and I will provide here some examples. And this is divided into two different areas, where the first one is more this engineering component, where we build tools to analyze big data. And then I will provide some discovery areas where we, in collaborative projects, work on discovery projects where we have to focus on certain types of questions, or we are focusing on certain types of questions that are relevant to us. Okay, then just as a preview of the most important data sets that I'm using in my research, it's always, especially when we combine genomics and drug discovery. So here is an example of the genome. And then we have these individual genes. And what is important to us, these genes, usually most of the coding genes are encoding proteins. And for drug discovery, I think it's an easy thing to understand is that small molecules usually bind to proteins and not the DNA directly. So the DNA pretty much provides the information to encode these proteins. And the proteins are then the doers. And for us, they are also then the target for drug discovery as to for identifying drugs that then perturb the functionality of proteins. They can activate them or inhibit them. More often they are inhibitors, and then using these small molecules to modify biology in a certain way. There are certainly also genetic ways of doing this. In this particular drug discovery-oriented way is we use small molecules for various reasons. It certainly has also more applications in human health because modifying the genome in humans as a therapeutical approach is possible already. And it's desirable in certain areas, but it's very limited to certain types of diseases and conditions. Okay, and then here are just a few examples that I'm bringing up. For the drug discovery part, we developed software that is reusable, shareable, and in our area, we often warehouse the software in community projects, where some of them are mentioned here. If you're not familiar with them, that's not so important. But what is important for us is that these resources are maintainable and expandable. So we can reutilize what we have developed 3 years ago. It is still useful, and we can work on something new. And the new part can then use those other components that we have developed in the past. And this way, the system grows in its application spectrum. Yeah, and then very often for those who work with software that is graphical, we often do two things. We have first software that runs on these clusters. Often they're very technical. So you develop software that is very command line driven. So it's really, and you see it in these science movies or action movies when people work on these black screens. That's exactly how it looks like in our case. But we certainly also develop utilities that are web-based and graphically oriented, and people can then utilize this who don't have these technical skill sets. Those run then on Amazon as well as local machines so that they can be utilized remotely within the community and are entirely public actually. And this would be an example of software that is linked to a technology. It happens to be here a database that was developed by the Broad Institute. And this is one of the most exciting databases for the parts that I will be presenting in a second, is a database where they have done a huge amount of drug treatments on human cell lines. And now we have over 200 human cell lines. This has to be often when we do things directly on humans, when we can cultivate these cell lines, we can much easier perform these experiments, because I would not necessarily like to take drugs that are not tested yet. So you want to do this in an experimental way that is ethically justifiable, and that's one way of doing it. The other way would be doing it in model systems like mice or smaller organisms, yeast and so forth. In this case, it's human cell lines, and they do systematically testing of over 40,000 small molecules. Most of the known drugs are included in this set of 40,000 drugs. And then they combine this with these high-throughput sequencing technologies. This one is actually not a sequencing technology, but a related technology to obtain the expression profile that is genome-wide for every single cell that they are testing here, and every single drug, a different combination. And it's very impressive. They have over 3 million expression profiles genome-wide for these individual drugs, as well as knockout lines. And so this would be comparable for those people who know about these technology to 3 million RNA-seq experiments. And that's for us a big discovery opportunity. I will show an example in a second, where we first have developed some software to make it more scalable for us to analyze these data sets. And so these are really huge data sets, and what we are doing here is to take these profiles of a drug that we are interested in and have then algorithms available that can identify similar profiles of all these drugs that are in the database, what profiles they have generated, and fish out drugs that induce similar profiles of expression changes of the gene. So expression changes often means usually a change in the gene activity to produce RNA. And this way we can identify similar molecules, but also molecules that we didn't know before are binding to the other target sites, usually proteins, that we didn't know before about. And this way we can make many new discoveries, and particularly in this case we work on a specific type of biological problems, and that's related to human aging. And so we have there certain sets of mutations in mice that induce some of these expression profiles, as well as drugs that are known to be anti-aging drugs, at least in mice so far. For some of them, we also have indications that they do similar things in humans. And we want to identify novel drugs that act as anti-aging drugs. And that's a big discovery platform for us, where we have developed some software. And then in addition to the software that I mentioned so far, we also develop workflows for these more complex problems to make the analyses more reproducible, and also have different user interfaces for users to work with these big data sets more efficiently. And then to the actual drug more application spectrum related to discovery projects. There we are very focused right now on problems in the human aging field and age-related diseases, where we are trying to address in a large consortium very fundamental questions related to aging. And we use there also genome sequences from different species, not just human. So we have genome sequences from humpback whales available, as well as different from mouse, for instance, which has a much shorter lifespan than humans. Then the bowhead whale. The most extreme example here is a clam that lives up to 500 years. These bowhead whales live over 200 years. And we are trying to identify certain components in their genomes that allow them to live longer. A challenging project, but very interesting. And to address these important questions, whether there are genetic components that program or pretty much pre-program how long we live. And there's certainly also what we know from epigenetics. There are epigenetic clocks in our genome engraved that determine, most likely also the health span of many humans and animal species. And then very often when you work in this area, what is certainly more important than simply extending the lifespan is actually extending the health span. So I think for many humans, it is more interesting to live a healthy life as long as possible. And then I guess personally, at least, I would rather die quickly when I'm not so healthy anymore. I definitely suffer from severe diseases, but have a healthy lifespan as long as possible. And actually that happens in some of these studies. We have access to centenarian populations, but many of these centenarians have never seen a doctor. When centenarians are people that reach a lifespan over 100 and are still alive. It's a feature in their medical record is that they don't become sick for a very long time. And that is certainly something we want to address, and we want to do this with multi-omics approaches. So we have the sequences of many of these organisms on the proteome, genome, and transcriptome level. And in my part of this big consortium project, we deal with the small molecules. So we perform a lot of comparative genomics and combine this with these large numbers of small molecules that are available to us. And then the questions that we are asking are which proteins in these organisms are perturbable by drugs, to mean accessible to drug treatments. Not every protein is easily accessible to be perturbable or influenced by a small molecule easily. And then we also, particularly for this healthy aging part related to drugs, we look more into drug repurposing than discovering novel drugs that are still experimental. The main reason is that it's for us a lower hanging fruit and more likely to work, with considering the approval process or also acceptance process in humans in general. No one wants to take a drug that you most likely have to take for a long period of time, most likely for your entire life, ideally, if it has this undesirable effect. So doing drug repurposing of something where we know the side effects very, very well from decades of drug treatment studies already is a much, much more easy approach. But this doesn't mean that we wouldn't go for novel drugs as well. And then another project that would be then connected to this is a healthy precision diets we are also working on. And these are just examples of opportunities that are now available with these large databases we have access to. These would be over 2,000 human pathways, biochemical and signaling pathways in the human genome encoded, and shows roughly in blue color here where those drugs we are interested in. There's roughly 200 drugs that are known to be anti-aging drugs. Some of them are very speculative, some are have much more experimental evidence, and we know where they are binding. And this helps us to identify where in those genomes the target sites are. And then with this expression profiling, we can then identify novel target sites in certain pathways we didn't know about, that they could become targets of interest as well. And this is just a big map of one of these results where we have data from human, from these cell lines, from this LINKS project that I explained before, as well as RNA-seq experiment. These are just an additional technology that is available from mouse and human. And then we also have proteomics data available as well And they are on a reasonable scale, so we have roughly 3,000 proteins that we are quantitatively profiling here as well. Where we are collaborating with some experts in Seattle who are doing this. And then the approach is pretty much this, by pulling out then small molecules that are ranking high across these different omics technologies. They become leads for us. We often are certainly looking for, are these known molecules or small molecules or drugs that are aging related are showing up at the top as well, but also new ones. And then we subject them to downstream experimental experiments, usually something that is relatively high throughput, like on certain cell lines, they are being tested, and later on they are fed to mice. Mice experiments take them much, much longer, and you are on a much, much slower scale if you work on a feeding experiment. And in this case, also one has to keep in mind that these are lifespan studies in mice, so you have to wait until you know whether a molecule extended their lifespan or not, and then you are in a 2-year range roughly when you do these experiments in mice. And then certainly what will be then the downstream outcome in 2 or 3 years, there should be a few candidate drugs that have been verified in mice, and that's why some of the drug repurposing is likely to be more successful, that we could start thinking about how to apply this to humans as well. And then this is a little bit connected to this project that combines a little bit my agricultural background with this more very biomedical approach here that is funded by the NIH. And so the idea here is that many of these small molecules are actually naturally derived. Many drugs that we know, probably 1/3 of the drugs that we are using and are FDA approved, have some relationship in their discovery process to natural compounds. And now we have access to very large databases from metabolomic studies as to what small molecules, including their structures, highly resolved, are available in different plant species as well as microorganisms and also organisms from the ocean. For this diet project, it's more important for the most part actually from plants. And then I try to identify the small molecules that are FDA-approved drugs and which of these drugs and which pathways are perturbable by small molecules that are present in plants as well as show up in our experiments with these lifespan studies and are of interest to us. And then the possibility there could be that you don't have to go to the drugstore, but you could go to the vegetable stand in your grocery store and make a diet that contains some of these small molecules in large enough concentration. And we are certainly also interested in diets that are being consumed by populations where we know and are very interested in why certain populations in Hong Kong, for instance, in Asia, in Japan, in certain areas in Italy or in the Middle East have a cluster of humans that easily reach a lifespan of over 100. And is this related to their diet? So it actually deviates from this expectations that most of these things are coming from genetic compositions, but how much can we actually do with drugs as well as with diets here in general? And these slides are showing then that these small molecules that we have found that are present in plants, what are their pathways that they are targeting? So what is the spectrum of activities that are actually plant-derived, and others are probably only accessible through synthetic compounds that have been developed for certain diseases, but we don't find necessarily in plants. So there we are a little bit dependent on what nature provides to us, and nature might not have over evolution developed certain small molecules that bind necessarily to certain types of pathways. So when we are very interested which pathways are perturbable by natural compounds that we find in those resources. Okay, that's pretty much all of the yeah, the main part of the presentation I had related to what big data is to us, as well as the research areas related to tool development for software, as well as some discovery. And just to close out here, I just want to mention, expecting that there's a lot of students in the audience who might be interested in going to grad school or to university in general as undergraduate students, that there are many programs at UCR that actually deal with big data or are related to big data sciences. And some of the examples are, for instance, computer sciences, plant biology, biology programs, biomed departments, and many, many others. So if you go to this list of graduate programs here, you find many of them. I cannot list all of them here on a single slide. And again, selfishly, I'm the director of a graduate program that handles a lot of big data as well, or teaches, trains students in big data analysis or big data sciences related to genetics and genomics. And that program is called Genetics, Genomics, and Bioinformatics, or short, the GGB program at UCR. And we also teach some classes there I'm involved in. And yeah, and that is the last part here, is just to acknowledge my students who work in my lab. So we have a lot of undergraduate students. We always welcome undergraduate students, and I often have many students who start as undergraduate students. That's often a very good trajectory for many of them, either to find a great job afterwards, right away, after they graduate, or to move on to grad school. And students who work in a lab, I think that's the best test for you if you like research or not. Go into a lab or a group like mine, which is more in silico, and work there, and then getting then accepted to a graduate program is, there's so many out there. You have to be realistic as to where you can go. It's competitive, but you can definitely find one, and including here at UCR. And then there's a lot of collaborators, external ones, especially with these big biomedical projects. We're working with a lot of different groups. And then most of the funding currently comes from the National Institute of Health, as well as from the National Science Foundation and some others as well. So that's all I have, and thank you for your attention, and I'm happy to take any questions. You want me to wipe it off?
I can do. So, a big thanks to Thomas Girke for this very clear presentation on big data, genome biology and drug discovery. For someone really working in that field, I realize how important this type of work is for the identification of the drug, tomorrow's drug. So I just want to remind everyone that if you have a question, if people attending the seminar online have a question, please post your question on the chat. I already have a few questions, but I also want to remind people attending in the audience, if you have any question, you're very welcome to raise your hands, introduce yourself, and ask your question. Does anyone want to ask a question in the audience, or should I go first? So I will go first to the question. So let me. So the first question from Karim Zaidi: does Dr. Girke and all other UCR data researchers participate in the NIH Allo of Us research project? If so, what type of research are being pursued? And I'm not familiar with this project, NIH project. Are you familiar with this NIH project? The Allo of Us, Allo of Us, ALLOFUS. I'm actually not familiar with that project. I don't know if you can hear Professor Girke. Should I give the microphone back? Okay.
All right. So the drug-related and health-related project that I've shown here that relates to aging, the main institute we're working with is the NIA. That's the National Institute of Aging, that is part of the NIH. Yeah. And they are, so a great resource there is that they have been funding for decades already, and that's like hundreds of millions of dollars of investments, these cohort studies. So these are patient cohort studies. And in our case, there are at least three or four of them involved. And in our case, they are centenarian studies. So these are where they have very detailed biomedical data, and these patients, I mean, many of them live at home and are fully functional and healthy, as I mentioned before, and they either come from a family background where it's known that something in their family is different in their genetic composition, so everyone gets insanely old. That is of interest, but also then having those from those families that are in an age range of 80 years, 90 years, 100 and 110 and beyond. When you get to 110 and beyond, then things are really shrinking in terms of how many patients you have available. And as you can imagine, this is a very, very, they take monthly, I believe, samples from them, blood samples and other things, cognitive tests and many, many other things they do. And all this data is available to us. And as well as all these omics components, like we have their genome sequenced. We have now for the proteomics, also proteomic samples for these 3,000 proteins available and many, many other things. So we are trying to combine here these clinical studies, we call them, with these omics technologies on a large scale. And that's handled through the NIA, yeah.
So thank you. So I have a few other questions. So we're sorry we're not familiar with this project, but familiar with the other question, I will say. So hello, my name is Swati Bala, and I'm a third-year neuroscience major and a science ambassador. I was wondering how personalized medicine and sequencing would be applied to cancer patients, and if that could be generalized to a larger population. So I guess.
Yeah, I guess definitely yes. I guess one can start more in the academic world. There is a big project funded by the NIH that's called the Cancer Atlas. And so we have large-scale sequencing data for cancer research. These single-cell sequencing approaches are also, booming like crazy right now because they provide so many opportunities to, take a cross-section of a cancer's tissue sample and sequence, every single cell and know where the differences are. And many cancers are often caused not necessarily by genetic material that we have inherited, but by somatic mutations. So mutations that we obtain during our lifespan. And that's what often makes the cancer genome sequence different from cells that are healthy from other tissues. Usually the genome is shared in our body for the most part across all the cells. So they have the same, in our brain we have the same genome sequence as in our liver or skin. And somehow during the lifetime there are somatic mutations accumulate. And certainly for certain cancer cell types or cancer types, there are many of them. The diagnostic has gone so far that in the past it was often ambiguous to diagnose a certain cancer and what type it is. And nowadays we use sequencing to classify what cancer it is. Sometimes they only need to sequence a short part of the genome, PCR it out, so this is a way of extracting DNA from a certain location very precisely, or sequence the entire cancer sample. And this Cancer Atlas actually provides a lot of information on the RNA expression profiles of these of these cancer samples. Proteomics is also starting, but again, it's more expensive and time-consuming. But the genetic background, meaning the genome sequence, is available for all these cancer samples. That often, these are usually human samples, so often it's a little bit harder to access those data sets because it's, when you use sequencing, then it is like a fingerprint that you could actually identify who the source is of these samples. So it often comes with certain, approval processes that you make a proposal to access those data, but it's not a big deal for scientists. But it's not like everyone in the public, like here, anyone in the Riverside area can access, the human genome and other things, or other genomes from animals. It's much, much easier. And that's totally publicly available without any string. It's like a Google search. You just need to know where to go. Yeah, I guess there's a little bit educational background in this area. It's a long answer.
Thank you. I'm aware of a patient sample being sequenced and used to actually administrate the right drug against certain certain cancer types. So yeah, I think the personal genomic medicine has already started and is certainly going to expand in the next few years. So another question from Karina Quevedo. What role does the epigenome play in drug discovery? Is it being investigated computationally? I think I can also answer this question, but what role does the epigenome play? Yeah.
So I guess this is a very booming area, and it's very exciting. The epigenome is pretty much something that is attached to our DNA, right? Parts of it sometimes are even inherited across generations, but for the most part it's not, and it's acquired over time. So in the aging research we are working in, I'm not involved in this one so much, but there's like very exciting publications that came out recently or in the last 10 years overall, that the epigenome plays a major role in setting a clock, the clock that I mentioned before, that might determine our lifespan, and that we at least by sequencing, so we have methods available to sequence the epigenome as well. We pretty much have specific sequencing methods that we sequence the DNA and have something associated with it that helps us to see what modifications along every single base pair are acquired and where those modifications are in the entire genome. And then we often want to do this also on the single cell level in those cancer samples and see whether there are epigenetic differences between cancer and non-cancer samples, and whether the cancer is maybe caused in some cases, or aging-related diseases are onset more likely or frequently because of epigenetic changes. And that is certainly a very interesting area, but it is, it's just at the beginning a little bit, yeah. Mm-hmm.
So we have so many very exciting questions. I think I will have time just for one more. And this is from Janet Franklin. Thank you for your talk, Thomas. Is it fair to say Earth's environmental science began to develop their big data science about the same time as the biological science? Example with the NASA MODIS that was launched in 2002. And what, if anything, have this informatic community learned from each other? So I think this is a very interesting question.
So earth sciences and biological sciences, their big data sets, whether they are, synergistic, maybe? Is that roughly?
So that they want
Yeah.
to understand if everyone has been talking to each other, and what have you learned from each other community?
I guess certainly it relates back to this more method development. We always exchange these methods, and they are shareable. So we use methods that physicists use, and we use in biology, and certainly also in earth sciences, and especially when it comes to all these resources. But I think the question goes more in the direction as to whether there are findings in the data themselves that are shareable across those scientific domains. I'm sure there are many examples, more specific examples, but I wouldn't have like a big example that would be particularly, so no, I don't have a good example. But I think it's an important question for sure.
Yeah, it's a very important question. I think we learn a bit from each other, and then we actually, both of this field, I'm thinking, have improved tremendously because of the improvement of all of this computational power. I mean, without computational power and these big computational cluster, none of us will be able to do this type of work. So I think, unfortunately, we don't have much time for additional questions. I don't know if we can actually keep those questions, and maybe Professor Girke will possibly try to answer some of those online if you're interested. But I would like to, we already over time to conclude this presentation on big data, the intersect of drug discovery and genome biology. Please don't forget, next week we should be back here. The science lecture series is back next week for the third big data science presentation with distinguished professor of mathematics Mark Albert. He will talk about computational modeling and digital twin of a patient. So if you like to watch Dr. Girke presentation, please feel free to go on the website. The video recording will be available on the science lecture series at ucl.edu and on the CNS YouTube website as well. And I would like to thank you all for attending this event. And again, thank you, Professor Girke, for this great presentation. And thank you again for joining us, and see you all next week.